Greetings. This video is about cell form and function. Now, this follows our discussion of macromolecules, which, as I hope you recall, are made of molecules, which are made of atoms. So we're moving right along our little path here. Now, cells are the first level that we're reaching where we're actually talking about living things. And cells are made of a huge variety of macromolecules, especially proteins. The variety of proteins is staggering. Okay, so as I hope you also recall, cells are the most basic units of life. All living organisms are composed of cells and their products, and all of a living organism's structure and function are due to the activities of its cells, even me talking to you and you learning what it is that I'm describing. Now, every cell has to come from a pre-existing cell. They cannot just appear out of thin air, which means that if you trace back the ancestry of every single cell in every living organism, there will be a common ancestral cell. And this is why all cells share so many similarities, use the same basic macromolecules, and so on. Now, a lot of what we know comes from looking at cells through a light microscope. However, it has its limitations, which I'll discuss. Now, light microscopes use a light source which shines through a specimen that would be on the stage and then is magnified through a series of lenses. These are called the objectives, these are called the ocular lenses, and these you will work with in the lab and practice the techniques for being able to view specimens clearly. The light microscopes that we use have a maximum magnification of 400 times, which seems like a lot, but a good light microscope or a typical light microscope will go up to about 1,000 times, and a really expensive one about 2,000 times. Still, that's not enough to see most of the things inside the cell. For seeing the parts of the cell that actually do the work, we need an electron microscope. Now, electron microscopes, as opposed to light microscopes, use electrons and bombard a specimen with electrons. The electrons have a much shorter wavelength, which allows them to resolve two things much better than light can. Light has a wavelength of 400 to 700 nanometers, and an electron has a 1 nanometer wavelength. And when we talk about resolution, we're talking about how in a light microscope, two objects will actually appear as one, but in an electron microscope, you will see them as two separate objects. Think about looking at headlights on a car from a distance. At a distance, it may look like one light source, but as the car gets closer to you, you're able to distinguish the two headlights separately. That's what the electron microscope allows us to do. So, light microscopes allow us to see the cell and its nucleus and sometimes some of the organelles. This is an image of a cell nucleus. This is the nucleus, and this is what's called the nucleolus through a light microscope. The electron microscope, looking at a very similar specimen, you can see how much sharper the image is of the nucleus and the nucleolus. So, a lot of what we know about the structure of a cell's organelles come from the electron microscope. Now, cells come in all kinds of shapes and sizes, even within our own body. Here we have the egg, the ovum, which is about 130 microns or micrometers big, and the sperm, which is about 5 microns or micrometers, so the biggest and the smallest. But they have different shapes. Some have projections, some have modifications of the membrane, some are, their shape is changing constantly, some are actually made of lots of cells put together. Regardless of the shape, they all have the same basic structures, and that's what we're going to go over in this video. Every cell is surrounded by a plasma membrane. The plasma membrane is composed of three main things: phospholipids, cholesterol, and proteins. Now, phospholipids we saw in the topic on organic molecules. These are fatty acids that are connected to a glycerol molecule, and then the third spot where a fatty acid might attach, if it were going to be a triglyceride, is actually occupied by a phosphate group. Now, phosphate groups are hydrophilic. They love water, whereas fatty acids do not like water. They are what we call hydrophobic or water-fearing. So if you take a bunch of these molecules and we say that these are amphiphilic, which means that they love both things, both water and nonpolar substances like oil. If you take a bunch of these and put them in water, they will spontaneously form a bilayer that then forms a sphere. So if we take just one small part of that bilayer, we can see that the fatty acid tails face each other and the phosphate head groups are on the outside, where they are facing and interacting with the water outside of the sphere or the cell, and also with the water inside the cell or the sphere. So what this means is that we now have a barrier that prevents most substances from crossing. So for instance, if there's a water molecule out here, it's like, oh, hey, phosphate groups, how's it going? Let me just squeeze through there. And it gets to the fatty acid tails and it's like, blech, and it turns around and leaves. Anything that's charged, sodium, chloride, potassium, can't get through. Same thing with molecules like glucose. The things that can get through are teeny tiny, like oxygen and carbon dioxide, or they are lipid loving, like estrogen and testosterone. Those can pass right through. The fatty acid tails don't bother them the least bit. Now, over here is an electron micrograph showing you the phospholipid bilayer. The resolution isn't terrific, but you can see that there are definitely two layers. And so what we're picking up here are the phosphate head groups, and you can see some of the lines of the fatty acid tails. This, by the way, is the nuclear membrane, which we'll talk about shortly. Now, the other thing, or one of the other things that's in the membrane is cholesterol. That's what these blue structures are here. Cholesterol is vital to the membrane because it gives it flexibility. So if it were just made of phospholipids, then the membrane wouldn't be able to bend. And we need it to bend sometimes in order for the cell to move. Cholesterol allows it to bend without rupturing or popping. We see a lot more cholesterol in membranes in cold areas because cold makes things more stiff. And so the cholesterol is needed to keep the membranes flexible there, even more than in warmer climates. Now, the last thing that I want to mention is membrane proteins. Now, membrane proteins have a bunch of different functions, and we're going to talk about a lot of those in a later topic. But just to give you an idea, some of these membrane proteins are there to move things in and out of the cell, like this. So substances will come in, bind, and then this will open up and let them go through, or the other way around. This one, because it has carbohydrates, that tells me that this is an ID molecule. So it's that's the way that it lets the rest of the body know it belongs here. This one is some kind of receptor. So when a signal coming from somewhere else in the body, a chemical, attaches to this, this will cause this little protein to detach and spread the word to the organelles inside the cell. Now, I want to point out one other thing that we can see in this image, and that's all of these red strands. This is part of the cytoskeleton. So just like our bodies have a skeleton made of bone that gives us the ability, shape and protection and the ability to move, every cell has a cytoskeleton that does the same thing. And we're going to see some better pictures of that in a little bit. Okay, now some cells modify their membranes with something called microvilli in order to increase surface area. On the left here, we see a thick band across the top of these cells. Just in case you can't see it, these cells are long and rectangular. You can kind of see the lines between them. These are the nuclei. And along the top, we have this thicker line that looks like somebody hit it with a highlighter. With the electron microscope, we're able to see that that thick line is actually made up of these little fingers. Villi means fingers, micro means small. And here we can see them even more clearly. Microvilli are there to increase surface area. So we see them a lot in places like the intestine, where the surface of the microvilli will have enzymes and transporters on it so that the food that's being digested passing through has more places where it can cross and get into the bloodstream. So microvilli are very small and used to increase surface area. Just to make this clear, the surface area without microvilli would just be this one line for this cell. But because of the microvilli that are going up and down here, we have a lot more surface area, which means that we can bring in more substances. Now, other cells have a structure called cilia. Cilia are hair-like structures. You can see them here with a light microscope. Remember, we couldn't see the microvilli with a light microscope, but cilia we can see. They're significantly longer. Here's a picture from an electron microscope showing the cilia. Cilia are not there for increasing surface area. Cilia are there to move substances that are on the outside of the cell. So we see this in a couple of places in the body. The respiratory tract, the cells there secrete mucus so that when you breathe in, the particles of debris in the dust that you breathe in gets trapped in the sticky mucus here, and then the cilia sweep the mucus up to your esophagus where it gets swallowed and destroyed, and this helps to keep the lungs clear so they can do their job. We also see cilia along the fallopian tubes or the uterine tubes of the female reproductive tract. So when the egg is released from the ovary in ovulation, cilia sweep it to the uterus. Without cilia, that egg would never make it to the uterus, which is where it needs to end up, especially if it's going to turn into a baby. Okay. This is a picture of the mucus on ciliated cells and the cilia sweeping to move the mucus along. So you get an image of what that looks like. Now, just want to remind you that microvilli are much smaller than cilia. So the magnification of these two tissues is very similar. And the microvilli are so small that we can't resolve the separate ones with a light microscope, but the cilia are longer and we can, and they're bigger. Now, there's one last membrane modification that we find in the human body, but it is only found in sperm in humans, and that's called the flagellum. Flagellum is singular, flagella is plural. This is a long structure that moves in a propeller-like motion that actually moves the cell, and this is what allows the sperm to swim. Okay, so inside the cell, we've talked about the plasma membrane. Now we're going to move to the inside of the cell. The liquid inside the cell, plus all of these structures that you see here, is referred to as the cytoplasm. If we just wanted to talk about the liquid by itself, it would be called the cytosol. Often these words get used interchangeably, but just so you know, they do have slightly distinct meanings. Okay, so let's start with the cytoskeleton. The cytoskeleton, here's a membrane, two sides of the cell membrane, and the cytoskeleton is actually made of 3 different kinds of fibers. You don't need to know the differences between these for this course, but you can see there are 3 different ways that proteins put themselves together to build these structures that go throughout the cell. This gives the cell shape and structural support. Otherwise, it would just be like a water balloon, and it would, gravity would give give it whatever shape, a flattened ball shape. But we need our cells to have specific shapes for their functions. Besides shape and structural support, it also gives cells the ability to move. So here you can see a cell called a neutrophil that's moving its membrane to basically swallow these three little red things. This is another cell that's growing its membrane, moving its membrane in order to move through tissue. And here's another type of immune cell that's able to move its membrane to make long projections to gather up things like bacteria. So without the cytoskeleton, this wouldn't be possible. This movement means that the cytoskeleton is rearranging itself, breaking itself down and reforming itself to make a new shape for the cell. It also serves as a highway or a network for all of the organelles in the cell. So every organelle is attached to the cytoskeleton in some way. And if we need this organelle, which is called a mitochondria, if we need it somewhere else, it can be moved along the cytoskeleton, the filaments, until it's arrived at the place where the cell needs that particular organelle. The reason I like these two images is because it really conveys the sense that the cell is full of cytoskeleton. A lot of pictures that you'll see just show a few little fibers, and you're supposed to imagine the rest. If you look at this one, the cytoskeleton has been labeled with fluorescent pigment, and it fills the entire cytoplasm. So where the pictures normally just focus on the organelles, keep in mind that every single one of them is nestled in this cytoskeleton, and that's what can move the organelles around. Okay, let's start with the structures now. The most obvious, and the only one really that we can see with the light microscope, at least the ones that we have, is the nucleus. Now, the nucleus stores DNA. That's what these strands are here, and we're going to learn more about that in the next topic. And it contains this structure or structures known as the nucleolus. We'll talk about that in a moment. Now, DNA contains the recipes for making all the cell's proteins. Each recipe is what we call a gene. So this is like a giant cookbook. If you read or hear that the nucleus is the control center of the cell, know that that is not the case. There is no control center for the cell, just like the cookbook doesn't determine what it is you're going to make for dinner. This is just where we store the vital information to do the work. If we had to talk about control of the cell, it would be much more like a total democracy without a real leader. Which is very hard for us to imagine, but this is how the cell operates. The nucleolus, or this dark section, here you can see it in an electron micrograph, is where ribosomes are made. Now, ribosomes are enzymes that actually make the proteins. So to sort of summarize this, the nucleus is where we have the cookbook, the DNA, that gives us the recipes for the proteins, and it is where the ribosomes are made in the nucleolus to be able to read those recipes and turn them into proteins. So we have the cookbook and where the chefs are coming from. So maybe this is like culinary school. Now, the nucleus, like the entire cell, is surrounded by a membrane, and this membrane has pores in it. You can see that in this electron micrograph, all of those little dots. It's sort of like a ball with holes in it. It keeps the DNA inside and protected, but the holes allow smaller things like ribosomes or RNA to get in and out easily. So we get the best of both worlds. Now, the next organelle that I want to tell you about is called the rough endoplasmic reticulum. Now, let's look at the word first. Endo means inside. Plasmic refers to cytoplasm. So this means in the cytoplasm. And a reticulum is a network. In fact, it means net. So if you do a Google search for reticulated giraffe, what you'll see is a giraffe where it looks like somebody threw a net onto it and then hit it with spray paint. So anytime you see this reticula, that means net or network. So this is a network inside the cytoplasm. And we call it the rough endoplasmic reticulum because when we look at it on the microscope, the edges of it look bumpy. Now, those bumps or those polka dots are actually ribosomes. So the ribosomes are formed in here in the nucleolus, which we can't see in this picture, and they find their way out and insert themselves into the membrane of the endoplasmic reticulum. Just so you know, ribosomes also come out here and just float free. And each kind of ribosome, whether it's on the ER or in the cytoplasm, makes a different kind of protein. Now, the rough endoplasmic reticulum is actually attached to the nucleus. So when the cell wants to make a protein, it will copy the recipe, copy the gene. That gene will come out into the endoplasmic reticulum, go through a ribosome, and the ribosome will read the recipe and make the protein. So you can see that a little bit here. So here's the nucleus, which is where our DNA is. We're going to copy the recipe to something called RNA, mRNA, and that's going to come out of the nucleus into the rough endoplasmic reticulum. It's going to find a ribosome, and the ribosome is going to read that, and on the other side, it will produce the protein. The protein then can get wrapped in this membrane that will form a vesicle, sort of budding off of the ER to move to the next organelle. Now, the endoplasmic reticulum actually has two parts. The rough endoplasmic reticulum is the one that's right next to the nucleus and has that polka dotted appearance. But coming off of that also is what we call the smooth endoplasmic reticulum. No ribosomes here. And it's also connected to the rough ER. If we look in this micrograph, here's the rough ER that has more the flattened sacs appearance, and the smooth ER that looks more like, I don't even know, fingers, tubules, something like that. Now, the smooth endoplasmic reticulum is where lipids are made in the cell. So that could be phospholipids, it could be triglycerides to store energy as fat. But if you're trying to remember the functions of these different things, remember that lipids, oils, and fats are smooth, they're slippery. So the smooth ER makes lipids. Now, the next structure I want to look at is the Golgi apparatus. So here it is in this diagram. And here's a bigger version of it. The Golgi apparatus makes carbohydrates and also helps to fold proteins. So if a protein is made by the rough ER, it buds off in a little vesicle, will come to the first layer of the Golgi, and the Golgi will help to fold it, and we'll put some carbohydrates on it. And then it'll move to the next layer through a vesicle, and the next, until that vesicle reaches the cell membrane and that protein can be secreted. Now, people say that the Golgi resembles a stack of pancakes, which takes some imagination, but you can kind of see why. It's just a bunch of flattened sacs, and they get smaller as you move towards the cell membrane or the plasma membrane, or bigger towards the nucleus. Now, if you're trying to remember what the Golgi does, but it makes carbohydrates. Carbohydrates are found in pancakes, right? Pancakes are made of starch, flour, and we pour syrup on them, which is sugar. Both of those are carbohydrates. So if you see something that looks like pancakes, think pancakes. And you'll remember that the Golgi makes carbohydrates. All right, the next structure I want to talk about are called mitochondria. Mitochondria, to me, look like little pill bugs, which I don't even know if you know what those are, but I used to play with them as a kid. Mitochondria are often nicknamed the powerhouses of the cell, and their job is to do really the most important steps in terms of converting the energy contained in a molecule of glucose into a molecule of ATP. Now, a lot of people will say that mitochondria make energy. They do not make energy. The only thing that makes energy, that produces energy, is the sun. What mitochondria do is convert energy from one form, glucose, to another form, ATP, which the cell can actually use. If we didn't have the mitochondria, we would not have enough energy to be able to do all the things that our cells do. Incidentally, mitochondria came from bacteria a long time ago that invaded a cell but didn't kill the cell. And they kind of worked out a deal where the cell was like, hey, I'll let you stay in here where it's safe, but you got to give me some ATP so that, I can do my job. So they have this cooperative relationship. Mitochondria, because they came from another organism, actually have their own DNA. And so this is passed on from the mother every time that we reproduce, because only the egg, that the ovary produces, has the organelles. The sperm does not donate any mitochondria. So there's some DNA that we only get from our moms. Now, making ATP it produces some pretty toxic byproducts. In fact, a lot of the processes that the cell does produce toxic byproducts. So we have these organelles in the cell called peroxisomes. If that sounds like peroxide to you, that's on purpose. They are able to make peroxide out of reactive species. And that really means taking a species like a superoxide radical, or I'm blanking, but something that wants to react with anything that it encounters. The peroxisome can take it and sort of neutralize it so that it doesn't do harm. It doesn't interact with our DNA or other organelles and make them stop functioning. So we see those a lot near mitochondria and also in organs that are responsible for a lot of detoxification. All right, the last organelle I want to mention is called the lysosome. Lysosomes, lys means to break, some means body. So these are sort of like the stomachs of the cell in a way. If we have an organelle, say this mitochondrion, and it's just worn out, right? It can't work anymore. It can be brought into the lysosome, and the lysosome has enzymes that chop it up into little bits that we can then reuse. So once we have those amino acids or the lipids, or whatever the basic molecules are, we can use those to build new proteins and structures. So they're like little recycling centers. All right, I think that's it.